So welcome back. So we were talking about the substances that are released from the cytoplasm of the platelet that have a key role in the activation of the platelet, and we made mention of 4 substances. We made mention of the adenosine diphosphate. We also made mention of what serotonin. We made mention of what the platelet-derived growth factor. We also made mention of what the platelet factor number 4. So now, how does these particular substances activate the platelet? So when these substances exert their effect on the platelet, we're going to see a change in shape of the platelet. There's going to be a change in shape from a disc shape to a spherical shape of the platelet. This change in shape of the platelet has a very, very significant role in the next processes that are involved in the platelet plug formation. And what are those processes, guys? We're talking about the platelet adhesion and also the platelet aggregation. So right now, also as a result of the activation of the platelet by these substances, there's going to be an appearance of these certain glycoproteins on the surface of the platelet. We're talking about glycoprotein 1b. This glycoprotein 1b, they will manifest on the surface of the platelet, ready to bind and adhere to the subendothelium collagen. So right now, what are the effects? What are the effects of the activation of the platelet? We said first, we talked about the change in shape from a disc shape to a spherical shape. We also talked about the appearance of certain glycoproteins. To be specific, glycoproteins 1b. So the whole of these changes is what is termed activation of the platelet. And this activation of the platelet helps the platelet to proceed into the next step of platelet plug formation. So what is the next step of platelet plug formation? We're talking about the adhesion. I want you to take a good look at this picture. In the platelet adhesion, you can see we're talking about here the activated platelet here. Because the platelet, before it can be able to undergo the adhesion process, it must be activated. So you can also see the cycle glycoprotein 1b that has been expressed on the membrane of the platelet. Okay, now let's discuss this in details now. Normally, whenever there is an insult to a blood vessel, this has an implication or an effect on the endothelial cells and damage the endothelial cells. So when the endothelial cells are damaged, what happens? The surrounding normal or undamaged endothelial cells start secreting a substance called the Van Willebrand factor, guys. You understand? It's abbreviated as VWB, Van Willebrand factor. Although you can also get the Van Willebrand factor is also found in the plasma, but a good number of the Van Willebrand factor that is involved in the hemostatic process is secreted by the endothelial cells. Okay, now this Van Willebrand factor, what it does is that within the damage, if you take a good look at this particular diagram, you see that within the damaged region that has been insulted, the Van Willebrand factor will now come and attach itself to the subendothelial collagen, because once the endothelium is damaged, we are going to have an exposure of the subendothelial collagen. Now the Van Willebrand factor will bind to the subendothelial collagen, and what next happen? The activated platelet, guys, will now come and bind to the Van Willebrand factor. When the activated platelet now bind to the Van Willebrand factor on the subendothelial collagen, that is what is now termed platelet adhesion. Now, what actually happened after platelet adhesion, guys? The next thing is what is called the platelet aggregation, which is the next step involved in the platelet plug formation. Okay, when the activated platelet now binds to the subendothelial collagen through what? The Van Willebrand factor. What happens next? What happens next is that what? Don't forget, the platelet, like we said before, they are enclosed inside a membrane. And within the membrane of this particular platelet, we also have what? Phospholipids. Now, the phospholipase A2, phospholipase A2 is an enzyme that we've learned from our normal biochemistry. From our biochemistry, we all as medical students, we learned this concept of enzyme from our biochemistry. So the phospholipase A2 will now go to the membrane of the platelet. And in the membrane of the platelet, what happens, guys? Phospholipase A2 is going to cleave arachidonic acid. And from those arachidonic acid, we all know arachidonic acid undergoes two pathways. We have the cyclooxygenase pathways and the lipooxygenase pathways, guys. Now, what we are interested in is that as a result of the metabolism of this particular arachidonic acid, which will now be broken down to the prostaglandins, and from these prostaglandins, we are going to get a thromboxane A2. Now, this thromboxane A2 is more or less like a perfume. Now, it is this thromboxane A2 that is going to be released from the platelet. And what is the main role of this thromboxane A2? This thromboxane A2 helps to attract more platelet to the point of the insult. Now, the thromboxane A2 attracting more platelet to the point of the insult brings about an increase in the population of the platelet within the site of the insult. When that happens, that is what is now termed the platelet aggregation. The coming together of so many platelets in order to line the region of insult where the vessels has been insulted. So right now you might ask yourself a question. Why can't the platelet bind to the normal endothelial cells? But within physiological state, when everything is normal, all things being equal, this does not happen. But why? Due to the fact that there are the endothelial cells, they secrete nitric oxide. And this nitric oxide helps to repel the platelet from binding. So it is only when the endothelial cell is damaged, that is when the platelet can bind. And it does not bind to the endothelial cells, guys. It binds to to the subendothelial collagen. But it is not only the thromboxane A2 that plays a role in the reinforcement of the platelet to the site of insult. There are other substances that play a very good role in reinforcing the platelet to the site of insult. A good example of them is we have the ADP. The ADP has a very important role, guys. It both activates the platelet and it also reinforces the platelet to the side of the insult, guys. Let's take a